Hello and a very warm welcome to everyone joining us for today's Select Science webinar entitled How to Measure Immune Cell Killing of Tumour Cells Effectively. My name is Carrie Haslam and I will be moderating today's presentation. I'm delighted to introduce today's expert speaker, Dr. Anne Lodge, Chief Science and Innovation Officer at Cellero. In the webinar, our expert will discuss different treatments such as CAR T cells, checkpoint inhibitors and adoptive T cell therapy. We will present data using tumor antigen-specific T cells from Cellero and widely available tumor cell lines to demonstrate specific lysis of tumor cells. We will also learn how the Incucyte Live-Cell Analysis instrument from Sartorius offers workflow monitoring of tumor cell lysis using fluorescent label of cytotoxicity. After the webinar, we will then move on to our question and answer session. So please feel free to ask questions for the Q&A session at any time during the webinar using the tab at the left of your screen. Simply hover over the purple speech bubble icon and click to ask your question. So without further delay, I'd like to hand over to our speaker and would like to thank her for presenting with us today.
And thank you, Carrie, for your introduction. A pleasure to be able to speak with a remote audience at this time and tell you about our recent studies in looking at cytotoxicity of tumor-specific T cells. Now, just to kind of set these things in context, at Cellero, we mainly provide characterized immune cells for discovery type experiments, also mouse cells, and a number of reagents that would be familiar to any immunologist. And we also provide contract research services on a small scale to assist our customers that may not have the resources or expertise. And it's because of these research services that we began to really look into better ways of measuring cell-mediated cytotoxicity. Now, this is the key effector mechanism of immunity. Whether it's a viral infection or a transplant or a tumor, a lot of what goes on to kill those cells is from T and NK cells that are equipped with cytotoxic granules to kill targets that trigger their various receptors. And it is this activity that's harnessed by the CAR T and CAR NK therapies that are making their way through clinical trials. By loading in a chimeric receptor, it's designed to direct the lysis to the necessary cells. And there's also drugs and biologics in development to enhance this particular function of the immune system. And so, naturally, what you need is robust assays to measure this. And the gold standard is the chromium-51 release assay. Been in use for many years and uses radioactive chromium taken up by the target cells and oxidized to then retain that within the cells. You wash off any chromium that's not incorporated, and add your immune cells or whatever effector, and incubate. After 4 hours or whatever determined incubation time, the medium can then be collected in the chromium-51 measured by a Geiger, but not a Geiger counter, gamma counter, that can measure that release of chromium from the dying cells. Controls are included to determine what the maximum release of chromium would be and minimum, and that's used to set your range of the assay. Then the data are expressed as a percent-specific lysis. And obviously, since it's been in use for many years, this works well. But there are obvious drawbacks. And the main one, at the top of the list, is radioactive. In fact, I almost just listed radioactive 5 times because this is the problem. It does emit gamma rays, and it has relatively high energy, so it takes lead shielding. Many of us that have been in the lab a while might remember shielding for phosphorus-32 that you could do with kind of a plexiglass shield. But this particular isotope is going to take 5 millimeters of lead to really stop it. And in spite of the fact that it biologically shouldn't be released from the cell, and except in the case of killing, it's been my experience that there's always a background, which can be relatively high in some cases, up to 20% of the label being released just spontaneously. So for these reasons, there's really a need for better assays. Now, I'm not going to cover all of these in detail. That would take much too long. And in addition to what I've listed here, there are certainly other methods in the literature, other devices, what have you. But these are some of the ones that we've actually looked at that I will touch on. Calcine labeling. I won't show any data of this, but this uses the same approach as chromium-51 labeling, but with a fluorescent label rather than a radioactive one. So there's a big improvement right there. But it suffers from the same problem as the chromium-51, and that is we've seen relatively high spontaneous release of the label, and that naturally cuts down on the dynamic range of the assay that's available. LDH is lactate dehydrogenase. This is an enzyme that's contained within cells, and there are kits on the market to measure the release of this. Now, since it's naturally present in all cells, death of your effector cells can affect the readout of this label. So that's a problem. When you measure the LDH, are you measuring the death of the tumor cell, or are you measuring some death in the effector cell that's been added? Luciferase labeling I will discuss shortly. Also, there are flow cytometric-based assays. And some of this, I'm sure, is just because immunologists use flow cytometry all the time, and it's a comfortable instrumentation for us. There are both, there's growing use of CD107A staining. This is a measurement of the granule membranes coming to the surface on the cytotoxic cells. So you're not looking at the effect on the target, you're actually looking at the degranulation of your NK or T cells or whatever happens to be present. We haven't worked with it very much, so I won't show any examples of that, but obviously you'll may see that used by others. And also kind of under the rubric of flow cytometric-based assays is labeling your target cells so that viability could then be measured by your cytometer. And I will show some examples of that. We've also worked with ATP-based readouts from the tumor cell. And of course, the live cell imaging is some of the data we most want to share because this is new to us and really exciting. Now, for luciferase to be used, the target cells have to be transiently or stably transduced with luciferase. Once they're obviously labeled. You can add your effector cells, and after 4 hours, at least in the case of the assays we looked at, we have collected culture medium in order to measure luciferase that's been released from the cells. Naturally, you need to add in ATP to allow that enzymatic reaction to occur. Alternatively, you can measure the luminescence of the co-culture, because as those target cells die, they don't have any ATP to generate bioluminescence, and that will decrease. So dead cells, no luminescence. And honestly, you could probably combine both of these methods, either measuring the medium and measuring luminescence of the co-culture. And this is an example of measuring the release of luminescence. In this experiment, we used the K562 NK target. This is a cell type that NKs will just naturally kill because they lack MHC class II antigen. We took that cell and transduced it stably with luciferase and ran an experiment here with adding NK cells and collecting the soup. Along this horizontal axis is increasing numbers of NK cells added to a stable number of targets to create different ratios, typically done. And then along this axis is the relative luminescent units. So it proceeds pretty much as you would predict. Target alone doesn't release any label, but we were able to detect the increase in luciferase in the medium over time. Of course, the shortcoming of the luciferase label is that it is sort of genetic. We're not as adept with that, but obviously if we wanted to label targets such as K562, that's handy. But what about all the other targets we might want to work with? It just gets to be a bit cumbersome to have to label to transduce and assure that you've got labeling of your target cells each time, or for that matter, maintaining a parallel bank of luciferase transduced targets. What we have done, at least in the case of non-adherent targets, is look at cytotoxicity by flow cytometry, as I mentioned. In this case, we can use carboxyfluorescein succinylmidyl ester to label the target cells. Now, this is widely used more often in proliferation assays, but it works very well also in cytotoxicity to just clearly label what's the target so that we can focus on those and ignore, gate out any of the T cells that might be present. After labeling, we add antigen, and you'll see that coming up here. In our case, it's always necessary. Adding the effector cells and then incubating overnight. After that point, we can add a viability dye and just run them through the cytometer. It's a very simple measurement of viability. And here's an example of what some of that data would look like. In each of these plots, the CFSE label is shown on the y-axis, and you can see that we're looking strictly at those that are labeled. And 7AAD, which is just a passive dye taken up that will bind to the DNA of the leaky dead cells. In this case, this is target cells with no effectors present. And you can see that they do have some cell death. Some cells have just naturally undergone death. But with increasing numbers of NK cells, you quickly see an increasing number of cells that appear in the upper right quadrant, indicating that they've taken up the 7AAD dye and have died. We go all the way from 30% death at roughly a 1-to-1 NK to target ratio to 90% death when there's a 10-to-1 ratio. And this is a relatively easy thing for us to run. In fact, sometimes we run it alongside our standard interferon gamma release assay. So on this slide, we have an example of measuring both interferon gamma and cytotoxicity from the same cultures. In this case, we're using the T2 cell line as a target. It's a very large cell, so we haven't even bothered to label with CFSE. It can be gated on simply based on its increased size over the effector cells. So what we do in this case is collect first the culture medium to measure interferon gamma, and that's shown in the lower two graphs. We've got cells specific for MART1 antigen on the left. This is common antigen of melanomas. And on the right-hand side, the WT1 tumor antigen, Wilms tumor antigen, which is found on leukemias and renal cancers and a number of other tumor cell types. And we're using both of these antigen-specific T cells. We always measure the interferon gamma output from our antigen-specific T cells, and we include that on the certificate of analysis. So if you ever purchase one of these, you'll have curves that look like these down below, where we're showing the interferon gamma released based on the amount of peptide antigen that's been added in. So the T2 cells are not expressing these tumor antigens, but we can add in the necessary peptide to sensitize them for lysis. So on the bottom here in the example with the MART1 T cells, in red is the MART1 antigen that we know is expressed by the tumor. And in green is a altered peptide ligand that has actually got improved binding. So you can see that because it binds to the MHC or HLA antigens better, it's recognized at a lower concentration of peptide. And we've included the WT1 peptide in here as a control. It's, there is no background in this case, either of interferon gamma release or target cell lysis. What's interesting here for the MART1 cells is that by adding in the altered peptide ligand, you can see that they will lyse the target cells at a much lower peptide concentration than they will release interferon gamma. So the cytotoxicity is perhaps a more sensitive readout for these cells. And that's true even with the native peptide, which is shown here. We're picking up at 1,000 picograms or 1 nanogram. We can see some level of lysis above background in this case. At the same concentration of peptide, we're really still at background. The same is true when you look at our WT1 T cells. Yes, they produce interferon gamma in the presence of peptide. But when you use that same peptide and you look at the lysis of those target cells, it's apparent at much lower peptide concentrations. So this is kind of two pluses in one package for us, that not only are we, yes, it's nice to see interferon gamma. We know this is a pro-inflammatory cytokine, so these tumor-specific T cells would presumably be great in killing tumor. But it's comforting, to say the least, to see that they are able to kill a target cell at a relatively low antigen concentration. So that's all really helpful, but we still have the drawback that it's not useful for adherent cells, and many tumor types are adherent, most of them. Aside from leukemias, you're going to encounter tumor cells that are sticking to the plate. Then you're looking at having to trypsinize them in order to run flow cytometry. And it's going to even then require significant flow cytometry time to acquire data on replicate cultures and plow your way through that, at least in our setup. Now, as I understand it, I'm older and not maybe aware of the advances in flow cytometry. Feel free to use flow cytometry. It works great. But we think we found something even better with live cell imaging. So here's the next example I want to share before getting to that live cell imaging is that we attempted another approach with our adherent cell targets. We thought, well, obviously, if they were luciferase transduced, we can see a decrease in that label. What if we looked at another label that we're familiar with, which is cell titer glow? It's a product from Promega. And we use it quite often to look at cell proliferation. But we decided to give it a try to measure a decrease in live cells. We used the Caspase cell line, which is a cervical carcinoma that expresses HLA-A2, very common HLA allele that many of our T cells are directed against. It also expresses the E7 protein of human papillomavirus. So this is kind of a nice target for us because it just so happens we have an anti-E7 specific T cell lines. In the case of this experiment, we used one recognizing residues 82 to 90 of the E7 protein and co-cultured fixed numbers of the Caspase tumor cells with increasing numbers of our E7 specific T cells. we added the peptide that they are specific for to half of the cultures because we weren't sure, what if, for whatever reason, we don't get good presentation of the epitope. So by adding in peptide, it has an opportunity to exchange on the cell surface with existing peptides in HLA. Many of you may have heard that, class 1 antigens have to come from inside the cell. But there's this little trick that we know in immunology where, yes, the natural presentation pathway is a protein inside the cell making it to the surface bound to HLA. But once HLA gets to the surface, there is an opportunity for peptides to dissociate and other peptides that are outside the cell to then bind. And that's what we're taking advantage of here. After incubation, the T cells are washed off, and we added cell titer glow, which gives you a luminescent signal that is proportional to the number of live cells present. So that's why the T cells had to go. We were only interested in measurement of the tumor. And this is an example of the data we got. We ran it more than once, but just as an example. In this experiment, it looked like adding the peptide clearly gave us a decrease in the amount of relative luminescence. And we said, great, this looks like killing all right. But in the absence of T cells, it was in the absence of peptide, in the absence of peptide, it was a little bit confusing because there did seem to be a decrease, but in fact, all of these bars are above the tumor targets alone. So what was clear is that we did have still some residual T cells present in the wells, and that kind of blows over or glosses over any loss of cells in those wells. So yeah, it might work, but we weren't really convinced. Which then brings us to live cell imaging. And we had seen this used in the literature, at the exhibits, and there are several systems on the market that allow you to image and capture data using fluorescent labels that looked really interesting to us. And, when all was said and done, after looking at our alternatives, we chose to use the Incucyte S3. And once you set up the imaging of the culture, you can kind of walk away and come in the next day, and the data is pretty much ready to analyze. And that really is attractive to us. And measuring the same culture repeatedly over time can allow you to reconstruct what that action is between the time you put the plate in the incubator and the next day when you come in. There's many things that can happen, as we all know, during that overnight incubation time. And it is very satisfying to be able to see what goes on in that black box during that time. Not to mention the fact that, at least in our laboratory, we're really visual people, so having the images is great. We had several reagent choices with our Incucyte system. The target cell labels can either be nuclite, which is would involve transduction of a plasmidien, and it would give us the ability to label the nuclei of the target cells. But we're still a little bit reluctant on transduction. We've gone with the Cytolite Rapid Red or Rapid Green. And we've titrated both of these labels, and we've seen very little toxicity until you get to the highest possible concentration. And we're able to, fortunately, start at a lower concentration. We've seen no effect on cell growth or viability. For the cell death labels, we have a couple of choices that have their pluses and best use cases. The Annexin V is a cell membrane label. Really, some of you may be even more familiar with this than I am. But what you were looking at is the cell membrane turning inside out. The Annexin V will bind to the inner side that is everted during apoptosis. The Caspase 3/7 reagent is not is looking at the activation of caspase, such that when the caspase is activated, it will cleave this reagent and release a DNA binding dye. So when using that label for cell death, you're going to get more of a nuclear stain. And I think I do have some examples to show you. Here's an example of killing of the Caspase tumor cell line with T-cell specific for HPV E7 protein. What you see are the tumor cells have been labeled with a fluorescent red label, the Cytolite Rapid Red. And they're obviously easy to pick out, even though there may be some, different intensity of label. The nice thing about the Incucyte software is you can set that so that it's recognizing these fainter red as well as the more intense red cells here in the field. These smaller cells that are just gray-black are your T cells. And there are 3 examples of cells that have been killed or set on their road to death. You can see that most clearly by this yellowish color, which is the overlay of the green. In this case, it's a caspase label. So the green color there within a red cell shows up as sort of this yellowish color. And so then you have 3 cells that are clearly expressing both red and green labels. You can also see up here in the upper right, one of the T cells is green. But because it's not red, we can exclude it from our analysis. So it's nice to see that right here in the center of the field are 2 T cells hanging on to a tumor cell that is on its way to death, is entered in the apoptotic pathway. So now when you put all of these images through, the nice thing is letting a computer figure out what's live and what's dead. And essentially what happens, to go back to this picture, images from the experiment are used to kind of set the parameters so that you're only measuring what events you want to measure. So it's similar to flow cytometry in that you can adjust your parameters to look only at large cells, only at small cells, just the red, thank you very much, or just the green. And that is done initially. And usually the parameters, once you've kind of worked them out, you don't even need to think about them anymore, but apply that analysis to every plate you run after that point. Now, I was about to show this. So in this case, we're looking at cytotoxicity of the AU565 tumor cell line, which is a breast cancer. And in this case, it expresses HER2. The T cells we've added to this are HER2-specific T cells. And we've let the experiment run for 18 hours and collected time points once an hour. So that part happens while we're comfortably at home social distancing. What you can see in this graph is that at the highest effector to target ratio, we've got great cell death. What's shown along the y-axis here for a readout is an increase over the starting point. So everything's been normalized in this case to the 0 time, and that's why everybody kind of lines up so nicely here at the bottom. And what we're measuring is the overlap object count in each well. So what you saw in the previous slide, it's only going to pick up those yellowish, those cells that are of the size that we've specified and that show both red and green pixels on top of each other. And you can set those parameters. So we've gotten just a count but then we've normalized with the starting point, and you can see that that particular effector to target ratio kind of bounces right out. Now, addition of peptide, different cell ratios also work, but you can see that those curves are definitely lower. And you can also see, because we've run so many time points, that if we had stopped the analysis earlier, we wouldn't nearly have the separation of the different conditions that we do in this slide. Now, the interesting thing with the Incucyte system is that you have control over what this readout signal is. In this case, overlap object count. But in this slide, we're still counting those overlap objects, but we've removed the normalization to the 0 time point. And the data begins to look a bit different. Now, naturally, at the beginning, there isn't much killing, but you can see just raw object counts. Those are actually higher in the presence of added peptide. Now, the data is still kind of makes sense if you compare it to the prior, but the question becomes, is it appropriate to normalize to the starting point? Or what I think we've settled on is, let's normalize it to the number of cells per well. And again, this is the advantage to using 2 labels rather than just one. We could have run this experiment just with the Caspase label, and we probably would have ended up with data that looks kind of like this. But I haven't gone back to reanalyze this with our more recent adoption, and I'll show an example in the next slide. The readout that I'm favoring at this time is an overlap object count divided by the red object count in each well. So if for some reason you've got a clump of cells in another well, those additional red objects, if you will change what the ratio of dead to live. It's nice to use this overlap divided by red, because you can imagine it's intuitively a percent killing. I haven't labeled it that way on the y-axis, but it's a way, I think, that allows some comparison between different experimental runs. That's harder to do if you're normalizing to a 0 time point. But in this case, we're looking at killing of a melanoma cell line by our MART-specific T cells. And so it's interesting to compare this to the earlier slides I was showing using flow cytometry. It is still showing best killing at the higher effector to target ratios. And what's interesting about this slide is we've also learned over time that it's important to have some sort of measure of irrelevant or bystander killing. And so in this experiment, we included T cells that are specific for cytomegalovirus, and those are shown in the blue lines. There is no cytomegalovirus in this culture, so the degree of killing we're seeing in these by those T cells is probably just background, and it seems to be about 8% in this example. And honestly, using other tumor targets and different combinations, that 8 to 10% may be about background. That, at least with these particular CMV T cells, there's some killing of the tumor cell that looks real at some level. And I can show you what that looks like in an image-based format below. On the left is a field of the melanoma cells with MART1 specific T cells and CMV specific T cells in the right-hand panel. What you can notice, even at this relatively low power look, is that there are a lot of T cells clustered around these tumor cells. but that yellowish color is more apparent here than over here. You'll also notice that there's an awful lot of green associated with the cells that have been added. We do see using that caspase label does show up a lot more cell death in the T cell population. Now, it may not be so much T cell death as the fact that they do express caspase. And so we're picking up a little of that. So here are some important considerations that we've run across with the experiments we're running. Establishing the T cell specificity using tumor cells, probably not the way to go, because it's hard to have both the presence and absence of antigen to be really certain about it. If you can take target cells and transduce the antigen in, that is ideal because you have the background antigens expressed by the target and then a known specific. We are looking at implementing some experiments looking at that. But we have really established the specificity of our T cells using target cells where we can add in peptide antigen. So we're pretty sure of our specificity of the T cells. Now, we feel we've now got a better handle on kind of a specificity control for the experiment. For proper interpretation of the data involves having some addition there that will give you that background lysis. The other thing that I found really interesting is addition of peptide can reveal poor presentation of some endogenous antigens. We know that over the years, we've accumulated a lot of information on what peptides bind well and which peptides even are, at what points they might be cleaved for creating that antigenic peptide. But there's still some gray areas there where it's not possible to predict an epitope. It's also helpful to just evaluate if you're going after a particular target antigen, is that thing really presented in a normal situation or not? And in some cases, we found that lysis of a tumor cell may only take place when you've added in exogenous antigen, telling us that, yeah, you can get recognition of that peptide, but on the target cell side, it's not seen. So we're having fun learning a bit more about our tumor cells as we go through. A few of the points for you all to take away is that there are multiple methods for measuring immune cell killing. Get out there in the literature and check them all out. Your choice will depend on what your lab prefers and what equipment you have available. As I mentioned, flow cytometry, many immunologists are comfortable with that. But I think that the live cell imaging gives us a way of doing flow cytometry in a static way. It allows us to leave cells right where they are and see what they're actually doing rather than, mucking about with the flow cytometer. But, others' choices may be different. The imaging, I have to say, can be very satisfying because what I didn't have time to show you or don't have a great example is the ability to put together little movies, watch your T cells crawl across the screen, watch as cytotoxicity develops. I mean, haven't been running these to make movies per se, but by creating those, you can see the process unfold right before your eyes. And there's not many things more satisfying than that. Even lay people like to see, look at this red tumor cell die because this tiny little tumor T cell is up against it and feeding signals in for death. So I know we have had great fun with our Incucyte, and I think our only regret is that we bought before the newer model came out with more colors, because more colors are always fun. But we look forward to generating more data with our instrumentation and sharing that with you via our website or even in our certificates of analysis. And I'd be happy to take questions.